Dear students, myself Dr. Mansi Arora, Assistant Professor in Biotechnology, Government College for Girls, Sector 14, Gurugram, and I'll be discussing with you the topic History and Evolution of Microbiology. Microbiology, as with any field of endeavor, can be comprehended best if one has a reasonable understanding of the historical development of the field. One cannot fully grasp the present theories and concepts without first understanding the logical steps that led to those ideas. Through this lecture, students will understand the importance of observations by Robert Hooke and Antony van Leeuwenhoek, theory of spontaneous generation and the experiments that dispelled it, contributions of several intellectual giants in the field of microbiology such as Louis Pasteur, Robert Koch, Edward Jenner, Paul Ehrlich, and Alexander Fleming. So coming first to the question, what is microbiology? It is the study of organisms too small to be seen by naked eyes. They are visible only under microscope. They include a large and diverse group of organisms that exist as single cell or a cluster of cells and include bacteria, archaea, actinomycetes, algae, fungi, viruses, and protozoans. Most of them are beneficial, however, some can be pathogenic also. Now, coming first to the discovery era. The science of microbiology started with the invention of microscope. One of the first to use a microscope was Robert Hooke in the year 1665. However, the magnification of the lens was just approximately 30x. But his unique contribution was the discovery of cell, which came through the observation of a cross section of a cork. He noted that there are compartment-like structures in the cross-section and called these cells. He published his work in a book called Micrographia. The first accurate description of microbes was reported in the year 1674 by Antony van Leeuwenhoek, a Dutch lens maker. He observed living microbes under a microscope designed and developed by him. He developed a lens system from ground glass that could magnify up to 300 times. With his microscope, he viewed rain and pond water, infusions made from peppercorns, and scrapings from his own teeth. He was the first person to accurately describe bacteria and protozoa, which he termed animalcules as he thought these were tiny animals. He also observed yeast, algae, spermatozoa, and RBCs. He reported his observation in a series of letters to the prestigious Royal Society of London, which published them in 1684 in English translation. Because of his pioneering contributions to microbiology, he is considered father of microbiology. He is also known as father of bacteriology and protozoology. The discovery of microbes spurred interest in the origin of living of things, but it also gave rise to some serious debate about the then prevailing concept of origin of life. Before 1860s, many scientists believed in spontaneous generation, that is, living organisms could arise spontaneously from non-living matter. Even the Greek philosopher Aristotle published writings that theorized how some organisms come into existence through non-living materials. Aristotle supposed that if a non-living material has a spiritual essence or vital heat, it could give rise to living organisms. The theory persisted for several years because without microscopes it did appear as though animals or insects were spontaneously appearing out of non-living things. For examples, fleas would appear out of dust particles. Fish would appear in a new puddle of water. Maggots would appear in rotting meat. Flies would appear out of manure, and mice would appear in a pile of corn husk or grain. Francesco Redi, an Italian physician, was the first person to challenge the spontaneous generation theory. In 1668, he performed an experiment to refute the idea that maggots, which are the larvae of flies, generate spontaneously on meat left out in the open air. Reddy placed meat in six beaker-like containers. Two were open to air, two were covered with some muslin cloth, and two were tightly sealed. extensive growth of maggots occurred in the meat that was left uncovered, but no maggots appeared in the tightly sealed jars. Although fly eggs were deposited on the muslin cloth over the covered jars, maggots did not appear on the meat. When ready, place the eggs that were deposited on the muslin on the surface of meats. The maggots quickly appeared. Hence, the experiment established the fact that the origin of maggots was the eggs of the flies and not the meat. However, John Needham was not convinced and had his own objections. In 1749, John Needham published a report of his own experiments in which he briefly boiled broth infused with plant or animal matter, hoping to kill all pre-existing microbes. He then sealed the flasks. After a few days, Needham observed that the broth had become cloudy and a single drop contained numerous microscopic creatures. He argued that the new microbes must have arisen spontaneously. In reality, however, he likely did not boil the broth enough to kill all the pre-existing microbes. Lorenzo Spallanzani performed a number of experiments that elicited results that contradicted those of John Needham. In 1767, Spallanzani boiled broth containing plant and animal matter. He then poured some of the heated broth into sealed containers and poured the remaining broth into unsealed containers. The broth that was kept sealed remained microbe free, whereas the broth exposed to air were found to carry microorganisms. Spallanzani's results suggested that the microbes found in broth samples that were exposed to air came from the microbes that were present in the air. Needham, however, argued that life originates from a life force that was destroyed during Spallanzani's extended boiling, and any subsequent sealing of the flask then prevented new life force from entering and causing spontaneous generation, whereas others argued that microbes were different from other forms of life. The legitimacy of the theory of spontaneous generation continued to be the source of heated debate until the mid 1800s and would have continued even further, but Louis Pasteur put an end to it with a very simple experiment. Pasteur made a series of flasks with long twisted necks known as swan neck flasks in which he boiled the broth to sterilize it. His design allowed the air inside the flask to be exchanged with the air from the outside but prevented the introduction of any airborne microorganisms which would get caught in the twists and bends of the flask's neck. If a life force besides the airborne microorganisms were responsible for microbial growth within the sterilized flask, it would have access to the broth, whereas the microorganisms would not. He correctly predicted that sterilized broth in his swan neck flasks would remain sterile as long as the swan necks remained intact. However, should the necks be broken or tilted, or the flask tilted so that the media came in contact with the microbes, the flask would get contaminated, allowing microbial growth within the broth. The next came the discovery of germ theory of disease. It was a belief that microbes cause diseases. Before this, the people believed that diseases were caused by divine punishment, poisonous vapors, curses, witchcraft, etc. As early as 1546, Italian scientist Girolamo Fracastro suggested that diseases might be due to invisible organisms transmitted from one person to another directly or indirectly. Oliver Holmes and Ignaz Semmelweis independently concluded that puerperal sepsis was transmitted by contaminated hands of obstetricians, nurses, and medical students. and this could be prevented by washing hands in antiseptic solutions such as chlorine water. However, such an important thought was not given much attention until the work of Joseph Lister. John Snow in 1854 advocated that the outbreak of cholera was due to the use of contaminated water from a certain pump. His assumption was found to be correct by discontinuation of water of that pump. Then came the studies of Louis Pasteur, and with this began the golden era of the microbiology. In the mid 1850s, Pasteur undertook a series of studies on alcoholic fermentation at a local distillery. In 1857, he presented evidence that all fermentation is caused by microorganisms and that specific microorganisms cause specific kinds of fermentation. Pasteur found that yeasts were responsible for converting sugar into alcohol in the absence of air, and the souring and spoilage were caused by bacterial contamination of the beverages such as milk and wine. Using his work with fermentation, Pasteur suggested that mild heating at 62.8 degrees Celsius for 30 minutes rather than boiling was enough to destroy the undesirable organisms without ruining the taste of the product. And the process was called pasteurization. Pasteurization was introduced into United States on commercial basis in 1892. His work on fermentation and pasteurization led to the development of germ theory of disease. Now, Joseph Lister applied Pasteur's work and introduced antiseptic techniques in surgery. Lister concluded that wound infections were due to microorganisms. In 1867, he used a dilute solution of carbolic acid to soak surgical dressings. Wounds protected in this way did not become infected and healing took place rapidly. So remarkable was Lister's success that the technique was quickly accepted and antiseptic surgical practice established the principles of present-day aseptic techniques. His techniques resulted in a dramatic drop in morbidity and mortality due to surgical sepsis. Because of his notable contribution to sterile surgery, he is considered as father of antiseptic surgery. Now, the undisputable proof of the germ theory of disease came from Robert Koch, a doctor by profession who was busy solving the problem of anthrax, which is a deadly blood disease in cattle and sheep in Germany. In 1875, he started experiments on mice. He injected mice with the blood of sheep and cattle suffering from anthrax. He noted that the same symptoms appeared in the mice. He isolated a few rod-shaped bacilli from a mouse's blood and placed them in the sterile aqueous humor drawn from an ox's eye. He inoculated the bacilli into the body of the cattle. The symptoms of anthrax appeared within hours. On examination, Koch found that their blood contained the same bacilli. He re-isolated the bacilli in sterile aqueous humor and re-inoculated into a healthy animal and found the appearance of the same times whenever he repeated the process. On the basis of these experiments and others on the causative agent of tuberculosis, Koch formulated a series of rigorous criteria now known as Koch's postulates for definitively linking a specific microorganism to a specific disease. Koch's postulate state the following. First, the disease-causing organism must always be present in animals suffering from the disease but not in healthy animals. The organism must be cultivated in a pure culture away from the animal body. The isolated organism must cause the disease when inoculated into healthy susceptible animals. And last, that the organism must be isolated from the newly infected animals and cultured again in the laboratory, after which it should be seen to be the same as the original organism. For his research on tuberculosis, Koch received the Nobel Prize in Physiology or Medicine in 1905. With the Koch's postulate as guide, Koch, his students, and those that followed them discovered the causative agents of most of the important infectious diseases of humans and domestic animals. These discoveries led to the development of successful treatments for the prevention and cure of many of these diseases, thereby greatly improving the scientific basis of clinical medicine and human health and welfare. Next came the discovery of laboratory techniques and pure culture. Pure cultures of bacteria were first obtained by Joseph Lister in the year 1878 by using serial dilutions of liquid media. With a specially constructed syringe, he diluted a fluid containing a mixture of bacteria until a single organism was delivered into a container of sterile milk. After incubation, the bacteria in this container were of a single kind, identical to that of the parent cell. Lister named the organism Bacterium lactis. Next came Robert Koch. He was carefully refining the methods for the study of bacteria. He found that smearing bacteria on a glass slide and adding certain dyes to them, individual cells can be seen more clearly with the microscope. Koch also concluded that bacteria could grow and multiply on solid surfaces and for the first time added gelatin to his broth to prepare a solid culture media. He used this culture media to obtain isolated growths of organisms known as colonies, each of which contained millions of individual bacterial cells packed tightly together. From these colonies, pure cultures could be transferred to other media. However, Koch did not like the gelatin he had used as a solidifying agent. It turned liquid at high temperatures and by chemicals produced by microorganisms. Fanny Hesse, wife of Walter Hesse, an assistant in Koch's laboratory, first proposed the use of agar in culture media. Agar is better than gelatin because of its higher melting and solidifying points. Petri dishes were also invented by an assistant of Koch named Julius Richard Petri in 1887. Thus, contributions of Robert Koch, Fanny Hesse, and Richard Petri made possible the isolation of pure cultures of microorganisms and directly stimulated progress in all areas of microbiology. Then comes the discovery of protection against infection, that is, immunity. Vaccination was discovered before germ theory, but it wasn't fully understood until the time of Pasteur. The first one to prevent smallpox by vaccination was Edward Jenner. During 1798, Jenner published studies on the immunization of humans against smallpox. Jenner observed that milkmaids who were routinely exposed to cows often developed pustules on their hands and feet that similar to those caused by the dreaded smallpox. Now, this cowpox was not fatal in humans, and apparently, those infected with cowpox did not contract smallpox. Jenner inoculated an 8-year-old boy with material from the infected cowpox pustules on the hands of a milkmaid. The boy became ill and had characteristic cowpox pustules at the site of inoculation but quickly recovered. A challenge dose later from an active case of smallpox did not result in illness, and the boy had apparently become immune to smallpox. Jenner had proved his theory that the pus in the blisters which milkmaids received from cowpox protected milkmaids from smallpox. He developed the first vaccination procedure but had no idea what caused smallpox or the mode of transmission. Now, Pasteur then worked in this field. Pasteur's first important discovery in the study of vaccination came in 1879 and concerned a disease called chicken cholera. It was chance observation through which he discovered that cultures of chicken cholera lost their pathogenicity and retained attenuated pathogenic characteristics over the course of many generations. He inoculated chickens with attenuated form and demonstrated that chickens were resistant to the fully virulent strain. Once he went on a holiday, and when he came back, he actually inoculated a bacterial culture which was left in the lab for almost 2 weeks into normal healthy chicken. And he found that they did not contract the disease. He was puzzled by these experiments. So, what he did was he again repeated the experiment with fresh bacterial culture and then immunized the fresh batch of chicken as well as those chicken who had previously been inoculated by the aged bacterial culture. He found that while the freshly inoculated chicken died, but those chicken which had originally been inoculated with the aged bacterial culture survived. So that is how he concluded that these chickens were resistant to the fully virulent strain. From then on, Pasteur directed all his experimental work towards the problem of immunization and applied this principle to many other diseases. In 1882, he attenuated anthrax bacillus by incubation at high temperatures and proved that inoculation of such heat attenuated bacilli in animals introduced protection against anthrax. He coined the term vaccine for such prophylactic preparations in the honor of Jenner's work. In 1883, Pasteur administered his first vaccine to human. He vaccinated a 9-year-old boy, Joseph Mister, who had been bitten by a rabid dog and helped usher in the practice of preventive medicine. The rabies vaccine was a new inactivated type of vaccine consisting of a neutralized agent rather than an attenuated microorganisms. And in this way, he laid the foundation of whole organism inactivated vaccines. Working in Pasteur's laboratory, Eli Metchnikoff discovered that certain leukocytes could ingest disease producing bacteria in the body. He called these special defenders against infection phagocytes and the process phagocytosis. He formulated the theory that phagocytes were the body's first and most important line of defense against infection. Paul Ehrlich applied stains to cells and tissues for the study of their functions. He reported that tubercle bacillus is acid fast. In 1910, he discovered selvracin, an arsenic compound, also known as magic bullet, that was capable of destroying the spirochete of syphilis. Now, this discovery gave rise to a new branch of medicine known as chemotherapy, and thus Ehrlich is also known as father of chemotherapy. He also studied toxins and antitoxins in quantitative terms and laid foundations of biological standardization. Mechnikov and Ehrlich were jointly awarded a 1908 Nobel Prize in Physiology or Medicine in recognition of their work on immunity. Later on came the discovery of viruses. In 1892, Ivanovsky removed the bacteria from diseased tobacco plants extract using some filters. This extract was still responsible for producing tobacco mosaic disease in healthy plants. In 1898, Bijring confirmed these findings and coined the term virus for such filterable infectious agents. And then many viruses causing different diseases in animals and humans as well as plants were discovered by various scientists. The discovery of the first antibiotic is credited to Alexander Fleming in the year 1928. He accidentally discovered penicillin produced by fungus Penicillium notatum that destroyed several pathogenic bacteria. He left his Staphylococcus culture on an agar plate for 2 weeks and went on a vacation. When he came back, he found mold on his plate which prevented bacterial growth. For this discovery, he shared the Nobel Prize in Physiology or Medicine in 1945 with Howard Florey and Ernst Boris Chain. Next comes the modern era, the 20th century, the era also of molecular biology. By the end of the 1900s, the science of microbiology grew up to the adolescent stage and had come to be known as a branch which is more inclusive in the field of biology. Many new discoveries happened in the biochemical, medical, and environmental microbiology. The microbiology became compartmentalized as several specialized subdivisions were made. In the later years, the microorganisms were picked up as ideal tools to study various life processes, and thus an independent discipline of microbiology, that is, molecular biology, was born. The relative simplicity of the microorganisms, their short lifespan, and the genetic homogeneity provided an authentic, simulated model to understand the physiological, biochemical, and genetic intricacies of the living organisms. Until this point, research was focused mainly on plant and animal cells, which are much more complex than bacterial cells. When researchers switched to studying these processes in bacteria, many of the secrets of genes and enzymes started to reveal themselves. And great breakthroughs were made during the later part of the 20th century, such as discovery of antibiotics, discovery of the DNA as genetic material, cracking of genetic code, identification of mechanism and regulation of gene expression, cellular immunity, RNA sequencing, host microbe interactions, as well as recombinant DNA technology. With the advent of genomics era in the last part of the 20th century, a number of techniques such as proteomics and metabolomics have developed, which along with bioinformatics has given a new thrust to microbiology. The studies are now focused on the application of microorganisms, particularly their genes, not only in the improvement of microbial products, but also the products of other organisms, including plants and animals. So this was the history and evolution of microbiology. Now, let's test your understanding by a few MCQs. Question 1. Who is known as the father of microbiology? A. Leeuwenhoek, B. Pasteur, C. Koch, or D. Lister? Question 2. Which of the following experimented with raw meat, maggots, and flies in an attempt to disprove the theory of spontaneous generation? A. Robert Koch, B. Lazzaro Spallanzani, C. Louis Pasteur, D. Francesco Redi. Question 3. Who discovered the importance of agar in culture media? A. Ehrlich, B. Petrie, C. Hess, D. Pasteur. Question number 4. Who discovered Mycobacterium tuberculosis? A. Virchow, B. Koch, C. Jenner, or D. Pasteur? Question 5. The purpose of swan-necked flask that Louis Pasteur designed to disprove spontaneous generation is to A. Allow the multiplication of microbes in the broth, B. Pasteurize the meat broth, C. Prevent air from entering the flasks, or D. Trap the microbes and prevent them from reaching the broth. Question 6. The event that triggered the development and establishment of microbiology as a science is the A. Development of microscope, B. Germ theory of disease, C. Spontaneous generation, and D. Use of disinfectants. Question 7. All of the following scientists got Nobel Prize for their contribution in the field of microbiology except A. Antony van Leeuwenhoek, B. Elie Metchnikoff, C. Paul Ehrlich, D. Robert Koch. Question 8. The father of medical microbiology is A. Paul Ehrlich, B. Edward Jenner, C. Robert Koch, D. Louis Pasteur. Question 9. The modern concepts of chemotherapy were proposed by A. Paul Ehrlich, B. Joseph Lister, C. Elie Metchnikoff, D. John Tyndall. And question 10. The term vaccine was coined by A. Robert Koch, B. Louis Pasteur, C. John Needham, or D. Edward Jenner. Here is the answer key for all these questions and the references for further studies. Thank you.